Hello, everyone. You're welcome to my YouTube channel. I'll be discussing the replication of RNA viruses. Viruses are obligate intracellular parasites. That is, they are known to depend on living host cells in order to multiply, to make more copies of themselves. These viruses are grouped into two: DNA viruses and RNA viruses. The replication of DNA viruses has been discussed in the previous video. And so, by way of introduction, viruses depend completely on living host cells for their replications. There are several steps in the replicative cycle of a virus, which include the following: the attachment to surface receptors on a susceptible host or susceptible cell, followed by entry into the cell, release of the viral nucleic acid, in this case RNA, followed closely by transcription of the viral nucleic acid and translation of messenger RNA for synthesis of virus-encoded proteins. The fifth step in the replicative cycle of a virus is the replication of the viral nucleic acid into progeny or daughter nucleic acid, then the assembly of the newly formed virus particles or virions, and finally release of the daughter virions from the host cell. There is an eclipse phase. It is usually the initial stage of virus replication, where the infecting virus loses its physical identity and most or all of its infectivity. During this phase, no virus is detectable in the infected host, and the eclipse phase is followed by the productive stage as new virus particles or virions are formed and released from the cell. The first step in the replicative cycle of a virus is attachment. Here, the viruses or the viral particles bind to the receptors on the host cell plasma membrane with their own receptor binding proteins. The virus receptors on cells could be glycolipids or glycoproteins. The interaction between the virus receptor binding proteins and the corresponding receptor on the host cell contributes to host specificity. A classical example is the hemagglutinin spike of influenza virus. The second step of the replicative cycle of the RNA virus is entry. After attachment, the virus gains access to the host cell environment where replication takes place. And viruses are known to employ three different mechanisms for this internalization. One, it could be fission of the viral envelope with the plasma membrane of the host cell. Two, receptor-mediated endocytosis of viropexis. Thirdly, direct introduction of the viral genome into the cytoplasm through channels in the plasma membrane. The third step of the replicative cycle of RNA viruses is the replication of the positive strand. Here, the positive strand of RNA viruses will have in single-stranded nucleic acid, a complementary strand is first of all synthesized producing double-stranded replicative intermediates. In piconaviruses, the single-stranded RNA acts directly as messenger RNA because of their polarity. The parental positive sense strand acts as template for production of complementary strand with negative sense. The negative sense complementary strand then acts as the template for the synthesis of the positive sense progeny viral nucleic acid. In single-stranded negative sense RNA viruses, for example, the rhabdoviruses, a complementary positive sense RNA is produced from the parental negative sense RNA. The complementary positive sense RNA then acts both as messenger RNA for protein synthesis. The proteins that are synthesized include non-structural and structural proteins, and also as the template for the synthesis of negative sense progeny viral RNA destined for encapsulation. Here is the flow of events during replication of piconaviruses. You have the positive strand parental RNA, which serves as template for the synthesis of negative strand, a complementary strand, which is negative strand RNA, and then this also serves as the template for the production or synthesis of positive strand progeny RNA. And the same positive strand parental RNA can serve as the messenger RNA for the production of structural and non-structural proteins which are incorporated, or the structural proteins are used for the production of the capsid that's around the nucleic acid in the progeny virus. Toga viruses and some other positive strand RNA viruses have a different replicative cycle from the piconaviruses, as shown in Figure 2. Only a portion of the genomic RNA is available for translation in the first round of protein synthesis. The probable function of the resulting products is to transcribe the genomic RNA to yield a full-length negative RNA strand. This negative RNA strand serves as a template for two size classes of positive RNA molecules. The first one is a small messenger RNA which is not translated in the first round. The polyprotein produced is cleaved into proteins whose main function is to serve as structural components of the virus, that is, they're assembled around the RNA as capsomes, forming the capsid. The second class of the positive RNA is packaged into virus. Here is the flow of events during replication of Toga viruses, which is quite different from that of the piconaviruses. You have the positive strand parental RNA, which can serve as a template for the synthesis of a complementary negative strand RNA, which also serves as a template for the production or synthesis of positive strand progeny RNA. And then the positive strand parental RNA can serve as the messenger RNA and is translated into non-structural proteins encoded in the 5' end of the genomic RNA. Here, the positive sense messenger RNA is synthesized from the negative strand RNA, and this is used in the production or synthesis rather of proteins encoded in the 3' end of genomic RNA. Now, both of them, that is the structural proteins produced here and the positive strand RNA, progeny RNA, are now assembled into the progeny virus. Now let's talk about the replication of the negative strand RNA viruses. The negative strand RNA viruses include the following: orthomyxoviruses, rhabdoviruses, paramyxoviruses, viruses. The negative sense strand viruses are divided into two groups. We have the multipartite group. This includes the orthomyxoviruses, viruses, and arenaviruses, while the monopartite viruses include the paramyxoviruses and the rhabdoviruses. Their genomic RNAs must serve two template functions in the first step for the synthesis of messenger RNA. And the second step for the synthesis of the complementary positive RNA strands, we serve as a template to make viral progeny genomes. The transcription of the viral genome is the first event after entry of the viruses into the cell. Here, the process uses functionally monosynchronic messenger RNAs, which are positive strands, each specifying a single protein. Replication then begins under the direction of newly synthesized viral proteins. A full-length strand is made and serves as a template for the synthesis of negative strand genomic RNAs. Here is a summary of the flow of events during replication of orthomyxoviruses, where you have the multipartite negative strand RNA serving as a template for the synthesis of positive strand RNA, which also serve as a template for the synthesis of negative strand progeny RNA. And the multipartite negative strand parental RNA can also serve as the for the synthesis of positive messenger RNA. That is, it is translated, it can be translated is transcribed rather to positive messenger RNA, and this is translated into proteins. Now, the proteins so synthesized, especially the structural proteins, are assembled around the negative strand progeny RNA and then leading to the assembly of the progeny virus. Here is the flow of events during replication of paramyxoviruses. Again, the monopartite negative strand parental RNA serve two functions. One, as a template for the production of positive strand RNA, and also can be transcribed into positive messenger RNA, which is translated to proteins. Now, the structural proteins so synthesized on the host ribosome are assembled around the negative strand progeny RNA, leading to the assembly of progeny virus. Replication of retroviruses. The third group of RNA viruses are the retroviruses. After infection, the sole known function of the genomic RNAs is to serve as a template for the synthesis of a viral DNA. The viron contains, in addition to the genome, an RNA-dependent DNA polymerase, which is actually a reverse transcriptase, as well as a mixture of host transfer RNAs, one of which serves as a primer. The key steps in the genomic transcription of include the transfer RNAs and also the synthesis of a DNA molecule, which is complementary to the genomic RNA, the digestion of the RNA by a viral ribonuclease. Actually, RNA is H, also packaged in the viron, which is specific for RNA in RNA-DNA hybrids. And then the third step is the synthesis of the complementary DNA strand and completion of a linear DNA molecule containing in its entirety the sequences contained in the genomic RNA, but with the duplication of two small sequences. One from the 3' terminus of the RNA duplicated at the 5' terminus of the DNA, and one from the 5' terminus of the RNA duplicated at the 3' terminus of the DNA. The double-stranded DNA is then translocated into the nucleus, where it is integrated into the host genome by viral proteins. The integrated viral DNA is then transcribed by the host RNA. The products of transcription are genome-length RNA molecules and shorter gene cluster-length messenger RNAs, which are translated to yield polyproteins. The polyproteins are then clipped to yield the individual viral proteins. The synthesis of at least one protein is accomplished by a ribosomal frameshift. Only the genome-length transcript is packaged into virons. Here is the flow of events during replication of retroviruses. Retroviruses with a parental RNA which serve as a template for the synthesis of a linear DNA, and then the linear DNA is integrated into the serve as integrated DNA, which can now be transcribed into positive strand messenger RNA and is translated into proteins. These proteins, structural proteins, are assembled around the positive positive strand progeny RNA, which are assembled to become the progeny virus. What's the significance of understanding replication of viruses? 1, for antiviral drug development. 2, candidates for vaccine development and applications in biotechnology. Here are selected references for further reading. Thank you for listening.